That's my fault. I keep doing this non-technical mistakes. Apologies for that. Okay, so continuing with the introduction part, where as I was saying, he's the member of the scrutiny committee, member of the Rajiv Gandhi Science and Technology Commission, Government of Maharashtra, and also at the national level, he is a committee member of BIRAC EUVA program. I guess many of you from the science fraternity is aware of this program, which is an initiative of Government of India, which supports innovation, entrepreneurship, and also development of different biotechnology startups. Based on or accepting his academic and research excellence, he has received several honors, especially the Achiever of the Year 2020, Creative Writer of the Year 2020, International Most Popular Article Award of 2020, Best Oral Presentation Award 2021, and the Presenter of the Year Award by Department of Biotechnology, Government of India. He is also a very passionate educator. When as soon as That's the main thing, I guess, that connects both of us. That I was really impressed with the way he approaches the education of new or budding biotechnologists. He has delivered more than, I guess, around 100 guest lectures and workshops at reputed institutions across India. He talks mainly about molecular biology, microbiology, biotechnology, and pharmacy. His hands-on training programs are dedicated to techniques in molecular biology, biomedical applications, and techniques in microbiology and allied sciences. He has received research grants exceeding 2 crore rupees, which has been funded by premier national agencies like ICMR, BIRAC, and DST, that is Department of Science and Technology. So these grants have been they have supported several innovative projects and advanced research initiatives, which has been led by Sir. As the director of Infinite Biotech, he has established a strong academic and industrial collaborations. He has around the firm, through his firm, he has around 50 MOUs, including some international collaborations with Saints University, Malaysia, University of Dodoma, Tanzania, and King Khalid University. I'm really happy the also to inform that we recently had an MOU with Dr. Mahesh sir, and we look forward to work together in the field so that both our arms, that is pharmaceuticals and biotechnology, work together at the innovation level and also at the training level. So it's all over to you, sir. So please continue with your session. Thank you, sir.
Okay. Thank you so much, Doctor, for nice introduction. So, can I start now?
Definitely, sir. Thank you.
Yeah. Yeah. Just a minute. So, screen is visible, Doctor?
Clear, sir.
Yeah. Thank you so much. Good afternoon, everyone. So, as sir introduced, so I'm Dr. Mahesh Khalate, working as a director of Infinite Biotech Institute of Research and Analytics, Sangli. So, today I'm going to cover some small part of the research. That's the animal tissue culture. So, from cells to solution, the power of animal tissue culture in modern research and industry. So, today we are going to interact with you people regarding what is animal tissue culture, how it works, introduction to the animal tissue culture laboratory, and some applications of animal tissue culture we are going to cover. And also, I'm going to cover the interesting part how you can start the animal tissue culture businesses using some government rules and regulations. So, first of all, what is animal tissue culture? So, as we know, so animal tissue culture is a part of the research. So, tissue culture is nothing but it's a in vitro maintenance and propagation of isolated cells and tissues and organs from the particular parental body. So we are going to culture the particular animal cells or human cells, rat cells into the lab using artificial conditions. We call it as animal tissue culture. Nowadays, the animal tissue culture plays a very important role in the research. So how it's going to help? So what are the requirements of the animal tissue culture lab, and what are the instruments required for to run the particular animal tissue culture lab? We will see in short. So first question. So why we need animal tissue culture? So because we know in research for testing the particular drug we need animals. So nowadays it's not possible to use animals in triplicate. Like so we are going to test a particular pharmaceutical drug or any particular API drug on particular animals. Nowadays in triplicate it's not possible. So that's why the animal tissue culture is a very knowing and very well known solution for the particular this problem. So we can use the animal tissue culture prior the test on the animals. So this is a very important requirement of animal tissue culture in nowadays in a modern research. So coming to the animal tissue culture part, so animal tissue culture means we are going to propagate the particular cells in a lab. We are going to use that particular cells for the analysis purpose. In animal tissue culture, we found three types of cells. We call it as a primary cell culture, secondary cell culture, and cell lines. Okay, so we'll see in short. So what is primary cells? Primary cells means so we are going to take a tissue from the parent, like any patient has a cancer, like a breast cancer. We are going to take a particular that tissue from the parent, and we are going to culture into the cells. We call it as a primary cell culture. So primary cell culture means you are going to take a cells from the parental body. We call it as a primary cell culture. So in primary cell culture, we regularly found the two types of cells. One is adherent and another is the suspension cells. So adherent cells means so cells they are sticky in nature. They need a surface for the growth in the laboratory. Suspension cells, we are going to provide a liquid media for the growth of the particular cells. This is the main difference. So adherent cells have some limit for the growth because they need a surface. Suspension cells, there is no any limit for the growth. If you are providing a new media to the particular cells, they will propagate easily. This is the main difference between the two cells in the primary cell culture. Depend upon the cells, you can select your medias for the propagations. So secondary cell culture, as I shown here. So first one is the primary cell culture. We are going to provide a new media, new flask for the that primary cell culture. We call it as a secondary cell culture. So primary cell culture you basically take from the parental body. Secondary cell culture you are going to take from the primary cell culture. This is the main difference. We subculture. Subculture means we all know. So subculture you are going to provide the particular new media, new conditions, particular for the growth of the cells. So second, the cell lines. So cell lines means so after the secondary cell culture, we call it as a cell lines. So because why cell lines? Because after secondary cell culture, cell have the ability to propagate more, as we call it as a finite cells and in continuous cell lines. Finite means we can particularly subculture that particular cells to up to the near about 300 to 400 times. But continuous cells, there is no any limitation for the subculture. You can propagate multiple times whatever you want. So this is the main difference between the cell culture and cell lines. Cell lines have the ability to propagate more than the cell culture. So coming to the laboratory part, so I'll introduce this how the animal tissue culture lab should be. So first, animal tissue culture lab should need the all these things like the laminar airflows, UV lights, because animal tissue culture is a very sensitive lab because there you are going to work with the cancer, very viral cells, any other the harmful cell lines. That's why we need the adapt the particular lab with the ultraviolet light, positive pressure ventilation units. Also, you can adopt some HEPA filters like the up to 0.22 micrometer. So it's provides the safe nature or a safe environment to work under the animal tissue culture. First, the important part, the lab, we have to construct the lab as per the particular standard protocol which is given by the government. So second part, coming to the instrumental part. So for animal tissue culture, first we need a biosafety cabinet. Everyone knows the biosafety cabinet, which helps to maintain the aseptic conditions in the lab while working. So first instruments, the very important. We need the biosafety cabinet. You may have the class 1 or class 2, depend upon the working. The second part, the CO2 incubator. So as we know, so for the culture shrink of the particular cell lines, we need some conditions like 37 degrees Celsius and some part of the CO2, 5% CO2. To incubate that particular cells, we need this incubator. We call it a CO2 incubator. Why CO2 incubator? Because we are going to provide the CO2 to maintain the humidity and the pH of the particular media. So that's why this incubator is called as a CO2 incubator. So basically, this incubator used to incubate the cells in the labs. So most important part, the microscopes. For this tissue culture lab, we need advanced microscopes like the inverted phase microscope and the fluorescence, confocal, whatever might be. So basic need is an inverted phase microscope because cells we are going to see from the bottom. That's why we need an inverted phase microscope rather than the compound microscope. So coming to the other instrument, the multiplate reader, might be the audience know this machine. So multiplate reader is basically used to read the particular observance of the reaction mixtures. After conducting the particular activity, we have to read the particular reaction mixture intensity. That's why we use this machine in the lab to measure the intensity of the particular reaction mixture. So most important, another part is cryocans. We call it as a cryopreservation in the lab. So cryopreservation is the very important factor in the animal tissue culture. You can store the particular important cells in the lab using this cryopreservation. You are going to provide a 196 minus 196 degrees Celsius temperature for the preservation of important virus cells or any cancer cells which are important to your particular work. So we call it as a, basically the cryopreservation. You can preserve the important cells in the lab. So coming to the another part, this is one of the small unit we need in animal tissue culture. We call it as a medium filtration setup. So medium filtration setup is required to filter the animal tissue culture media, which we are going to provide for the cell proliferation. So other consumables, basically we need all we I might be know about these all things, though 96 well plate, 24 well plate, some cell culture flask, and some automatic pipettes. So basically we will require to start the animal tissue culture labs at a particular point of the business. So these are the basic consumables you should need while working in animal tissue culture. Yeah. The main part when we are going to start the animal tissue culture, how you are going to isolate the cells from the parent body. If you have some particular cells in your hand, how you are going to culture or isolate a particular cells from the parental body. Here, I listed a small process how you are going to isolate the cells from the parental body. So here, uh, we we are seeing here's one is a liver tissue. After that, we collect the liver tissue from the particular hospital or any other patient, or you need a particular permission to collect the tissue for any cancers or virus tissue from the government. So after taking the tissue, you can utilize your lab to culture the cells. First, we have to break that tissue to take out the cells from the particular tissue. That's purpose. We have two methods. One is the enzymatical and second one is the mechanical method. So enzymatically, you can use a trypsin enzyme. We all know trypsin acts as a detaching agent. It will break the tissue when we are using in higher concentration. So use the trypsin. After that, break the tissue using some homogenizers. You can culture the cells in a particular tissue. Just a minute. Sorry. So particular cell culture flask. So after that, we call it as a formation of monolayer. Monolayer means so cells adopt the particular flask into the flask. They will cover the flask. We call it as adherent cells. After the formation of monolayer, you have to subculture the cells. So after subculturing, we call it as a primary cell culture, secondary cell culture, and tertiary cell lines. This is a small process how you are going to isolate the particular cells from the particular parental body. So we can use a trypsin enzyme, otherwise you can use the mechanical methods like tissue homogenizers, and you have to provide the media for the cell culture. That's a simple process how you are going to isolate. So I given here one example how you are going to isolate the brain tissue or a brain cells from the mice into the lab. So first we are going to take the mice, we are going to dissect it and up to the brain part. After that, you are going to section the particular brain part. You are also cutting the brain part, and after that, you have to chop using a homogenizer or any choppers, and put it into the particular cell culture, slice into the particular cell culture media, and you can incubate into the lab. So after 3 days, you get to know whether cell is proliferating or not. So basically, this medium is required for the cell culture proliferation. We call it as the Eagles medium. So many medias are there. So you can vary your constituents in the media like the fetal bovine serum, sodium bicarbonate, streptomycin. So all things you can vary while using this particular cell culture medias. So how we incubate the cells into the lab. So we use a particular cell culture flask as seen in the image or a slide. Uh, we are incubating the cell culture one by one of this is a standard protocol how we are going to incubate the cell culture. These are the flasks. We call it as a cell culture flask for the incubation of the cell culture cells or a cell culture we call. So particularly we call this is as a incubation of the cell culture into the lab. So main part of animal tissue culture. Doctor, you are seeing my slides. I think some network issues going on here.
No,
Any
no,
slide?
slides are clear, sir.
Slides like here?
Yes,
Oh, sorry for the
clear, sir.
inconvenience.
Incubation of cells with required conditions, right?
Yeah.
Yes, yes. Yeah.
Just a minute. Give me just a minute. Sorry for the inconvenience. So I'm facing some network issues today here.
There is some power issue also. Since morning, there is no component.
That's not a problem. Just a minute. Give me just a minute. Okay. Sorry for the inconvenience. Now the slides are clear. So this is on just a minute. Doctor, so the slides are visible?
Yes.
Yeah.
Now,
Sorry for the inconvenience. There here some network issues are going on. Okay. So in animal tissue culture, we call it as a passaging of cell lines. So passaging means we are regularly subculture the cells to avoid the contamination, some other because sometimes cells also need the new media, new nutrients for their proliferation. So we call it as a subculture. So basically, you can follow this protocol to subculture the cells. But that's a very simple step. You have to take the flask, add some PBS, phosphoropersaline, to maintain the pH. Again, add a trypsin to detach the cell from the surface, centrifuge it, and add a particular live cells. After centrifugation, you will get to know whether the cells are dead or not, because dead cells are very uh, clear in nature because they are floating into the media, and the live cells at the bottom they are high in their weight, and we can separate that. We can add a new media to the particular cell culture. This is, we call it as a passaging of cell lines. So, what are the environmental effects on the proliferation of cell? So, there are many environmental effects. We can call it as a or a biological effects we can call. So pH also matters. We have to maintain the very good pH for the proliferation of cells. CO2 also matters. We have to maintain the 5% CO2 atmosphere like the 37 degrees Celsius, any humidity required for the particular growth of the cells, and also the temperature 37 degrees Celsius. These are the factors we have to maintain the very clearly for the proliferation of the cells. So just a minute for that. Sit. So coming to the application part of the animal tissue culture. So there are many applications in animal tissue culture as we know as a researcher. So we will see one by one. So we can use animal tissue culture for the toxicity testing. Whether you are working on a particular novel formulations, you can use this animal tissue culture for whether the particular formulation or a drug toxic in nature or not. We can utilize this particular animal tissue culture for the toxicity testing. Cancer research, very well novel application of the particular ATC, animal tissue culture, is the cancer research, because we can analyze the particular novel formulation whether it's working on particular cancer or not. So this is the main application of the animal tissue culture. We can use the ATC for cancer research. So another is a major, the lab grown meat in North India. So US already given a permission to for the lab grown meats already in market into the market. So we can use this animal tissue culture for the preparation of lab grown meats uh, using animal tissue culture, and we can also market as that. But in India it's difficult. But other countries like USA, Europe countries, they already given permission for the lab grown meat. So coming to the main application of the animal tissue culture, so cosmetics. As we know, so all cosmetic products which we are regularly using, so we don't know whether the cosmetic product are irritating or they might be the toxic in nature. So all the cosmetic product we can check into the animal tissue culture lab for the irritation test or a toxicity test. After passing the in vitro, we can go for the in vivo section. So this is one of the major application. Coming to the next part, we can also use the animal tissue culture for the preparation of some tissues or like any this like the skins, because nowadays we know, so in urgency we don't hire or take any particular skins, required skins into the any hospitals or market. So we can use this ATC for the preparation of skins like any other organs using the 3D bioprinting. The in this particular part the research is still going on, so we can in future might be it helpful for the society. So coming to the research part, so in animal tissue culture, we regularly in Infinite Biotech, we doing the cytotoxicity assays to check the particular toxicity of the samples like extract, plant extract, apiatrix, or any derivatives. So these are some assays we are regularly screening in Infinite Biotech. We can call it a trifon blue assay, DNA fragmentation assay, MTT assay, or a proliferation assay, wound healing assay. We are regularly doing in Infinite Biotech. So these all are assay used to check the toxicity of the particular drug. Today I'm going to focus on one assay, that is the MTT assay. So how its particular activity going to work for the toxicity test. So for animal tissue culture, uh, checking a particular cytotoxic or anti-cancer assay, we use a method, we call it as a MTT method. So it's basically a 3 days procedure. We have to follow these 7 steps to analyze the particular drug for the anti-cancer or a particular cytotoxicity. So steps are imaging. You have to image the cells, media preparation, cell counting, seeding, drug addition, reagent addition, and then results. We'll see one by one in brief. Okay. First, when we are going to start the particular assay for the cytotoxicity testing, we have to analyze the cells under the microscope, whether the cells are live or not. So, what is the number of dead cells? What is the number of live cells? Because testing, we need more number of live cells. That's why imaging is very important. In imaging, we get to know whether the live cells are working properly or not, whether the contamination is going on, any fungal contamination, bacterial contamination. The only one solution, we have to image the particular cells before starting the assay, because in activity we need a more number of live cells. Okay. So after imaging, why imaging required? That's I explained. So to differentiate the live and dead cells, and also the to get to the percent of viable cells, to observe the any unwanted contaminations. If the contamination is there, we are not going to use that cells for the activity. After that, the purpose of imaging. So as I see shown the particular images of the cell culture. So here we can get to know whether the live cells or dead cells. In first picture, we can see here. So some white dotted burst cells. This is the dead cell. These are also some dead cells. So the oval shaped cells are the live cells. So here I get to know whether the live cells are more or not. So if the live cells are more, then only I'm going to use that particular cells for the activity. So after that, so you have to use the media, this media, as I earlier told. So this is a very important media uh, to do the activity also. So before taking the cells, you have to also provide the media to the cells. After imaging, you have to count the cells because we want exact number of live cells. So basically, we know that we use a Newark chamber to count the cells. We use a trifon blue dye to count the cells. Here some images, uh, how we are going to count the cells. Uh, Here some difference that we can see. So there is a total black dot. We call it as a dead cells because dead cells don't have any capacity to oppose the strain, and only the border strain we call it as a live cell. So we can differentiate easily. We can count the cells into the newborn chamber. Okay. After that, um, you have to seed the cells. Seeding, we normally seed into the 96 well plate, which is very easier to seed. Because what is the purpose of seeding? Because purpose of seeding, we are going to take the cells for the activity. So we are not going to add the whole drug into the flask. We are going to take the cells out for the activity into the 96 well plate. We call it as a seed. So after seeding, you are going to add the particular test sample. So on the particular well, you are going to add the various concentration of the test samples. After adding it, you are going to incubate into the particular cell culture incubator. So on third day, you are going to add some reagents. We call it as a MTT. So MTT plays a very important role in detection of live and dead cells. We will see next slide how it's working. So here I shown. So basically MTT in yellow in color. So when we add into the particular MTT into the on a live cell. So live cell has the live mitochondria. Also the live mitochondrial dehydrogenase enzyme. It will redact the MTT and convert it into the purple formazole. So it indicates that the more intense of purple color has the more number of viable cells. So if there is any yellow color, well, we can tell it, you can call it as a, there is a more number of dead cells. So this is the mechanism of MTT, how MTT plays, because when we are going to add the MTT on a live cell has a live mitochondria, also the enzymes. That enzyme is going to redirect the yellow MTT into the purple chromosome. So purple color means there is a more number of live cells, and yellow color means there is a more number of dead cells. That particular color intensity we are going to use you are going to use the ELISA reader to measure the intensity of the particular reaction mixture. So after reading the observance, we will use this formula. Basically, everyone use in biomedical research the percent of inhibition. We are going to calculate whether the cells are dead or not. There is more number of percent of inhibition. There is then that particular drug act as a good in anti-cancer. There is a more number of percent of probability. Then that particular drug going to act as a cytotoxic drug. So this machine we use regularly to measure the observance of the reaction mixture that we call it as the ELISA reader, also the microplate reader. This is a 96 well plate. We seed the cells into the particular wells, also drug, then MTT. After that, we are going to read into this microplate reader. So as earlier we discussed, this is the mechanism of MTT. So when we add MTT on the live cells redirect the MTT into the purple formation. Here one example. So you can see there is a high intense color of purple. There is a 0% of inhibition. Whenever we are going to upstairs, there is a change in the color. So here we can see the intense yellow color indicates the more number of dead cells. So this is the recall of the assay. So what is the recall? So this is actually 3 days procedure. First day we are going to image the cells, then count the cells, then seed the cells. This is the first day procedure. Here, second day procedure, you are going to add the drug and again incubate. Third day, adding MTT and reading a particular observance using particular ELISA reader machines. After that, you can calculate the percent of inhibition using the percent of inhibition formula. So also the major application of animal tissue culture is study of apoptosis. As we know, apoptosis is playing very vital role to eliminate the dead cells from the body. So also we can study using this animal tissue culture, the apoptosis studies, whether the particular cells acting as apoptotic in nature or not. As we know, the cancer cells are not apoptotic in nature. They are not going to die because due to their loss of genetic material, mutation into the genetic material, they don't have that capacity. In that time, we can apply the extra drug on cancer cells. We can convert that particular cells into the apoptotic body using animal tissue culture. Also, this is a small part. I'm not going to uh, cover this because this is practical part. So also we can use the animal tissue culture for the in vitro wound healing assays. As we know, though after a diabetic patient don't have that capacity to heal the wounds as much as earlier. So we can use this animal tissue culture lab to study the in vitro wound healing assays. How these are some protocols I'm not going to cover because this is all total practical part. So we can use this ATC for the particular wound healing assays, how wound healing happening or not. So what is the extra drugs we can add, manipulation of the drugs we can study using this particular animal tissue culture lab. So you can, I'm going to explain some part of the results. You can see here in the image, the first day we made the scratch into the particular cells. In second day, we add the particular drug. After particular hours, we are going to image the particular cell lines whether the healing means the area between these two scratch is nearest or not. If the cells are coming nearest to each other, it means the particular cells have the wound healing property after adding the drug. Though this is a small assay we can perform into the lab. So whether drug acting as a wound healing drug or not. So coming to the specific applications of the animal tissue culture, we can use the animal tissue culture for the production of many vaccines, propagation or multiplication of important viruses which are important for the research, production of monoclonal antibodies, production of therapeutic proteins, production of biopesticides, gene therapy, cell therapy. Many applications are there. I listed out a very few here. So nowadays, animal tissue culture has a lot of scope in research. As a coming to the modern research, animal tissue culture is nowadays playing a very vital role into the research. So coming to the specific business part, in India we have a lot of animal tissue culture in animal tissue culture, private institutes in cancer research, vaccine manufacturing, gene therapy, recombinant proteins, drug selection and improvement. We can use R&D, stem cell biology, IVF technology. As a business point of view, we can see the importance of animal tissue culture also. So this is a small part of animal tissue culture, how animal tissue culture works. So second session, a second part we can call, I'm going to cover how you are going to use animal tissue culture as a part of bio business. So how you are going to set up a business help with the help of the animal tissue culture. So we will see in short. So we call it as this particular science part we call as a bio entrepreneurship or a bio entrepreneurship. So anyone, so from the audience, do you know what is the bio entrepreneurship? Anyone? Anyone has the idea of what is bio entrepreneurship? Anyone from the audience? Do you know what is the bio entrepreneurship or a bio entrepreneurship? If not, I'll be short. Okay, no problem. So we all know the entrepreneurship. Okay, in entrepreneurship, the owner and manager are the business risks. In bio entrepreneurship, the owner and manager are there, but we are playing with the scientific risks. We are going to introduce the science into the particular business. We call it as a bio business. Okay, so as per the report of 2023 in India, so most of the businesses started in research and development. So in research and development, we basically has the particular ATC lab. So near about the 41% businesses started in animal tissue culture as per the report of 2023. So these are some sectors we can use the animal tissue culture for the uh, flexing of particular businesses into the market. So today I'm going to cover some small part. So how you are going to convert your ATC ideas from lab to market. Here I am showing some small journey. If you have an idea in your mind how you are going to convert it into the product. So this is a small pathway. So we all have to follow this pathway. So first, if you have an idea, so we call it as a lab. So first, you should have idea. We call it as a brainstorming idea. If you don't have idea, we are not going to work into the research because there is no idea, there is no solution for particular problem. So you should have a unique idea. You are not going to copy anyone. You should have your own idea. So if you have idea, so you can develop the product from the particular idea, but you have to follow the rules and regulation of the government. Also, the IPR also comes into the particular action. And what is the need of the particular your ideas or a product into the market? You have to evaluate using your particular areas or you can particular state you can call. So for starting a business, you should need a small scale laboratory, or if you are starting a business into the research and development, you should need your small scale or a large scale your own lab. So I will going through how to set up a small scale laboratory innovation labs using a government authorities, or you can also invest your own. But nowadays it's very difficult to invest our own money because instruments are very high costly in nature. So if you have interest to how to set up a small scale laboratories, so first we have to design the lab as per the standard protocol. We have to follow the government rules and regulations. You can start designing the lab, selecting the space or a commercial space, any residential space you can use, but depend upon the particular your business. If you're doing a business in the research and development, I suggest use a commercial areas to start the business, and after that design the lab as per the standard protocol which is given by the government. After that you need instrumentations. You need instrumentation for start the lab, but nowadays it's not easy to buy the instrumentations because all the instruments are nearly about in crore lakhs. It's not easy, but it's not impossible also. We can raise the funds to the government. So I'll we will see next slide. To start the any particular private or a semi-government laboratories, you need these all the what we can call license. So you should register your company with the Clinical Establishment Act. We can call registered with the Medical Establishment Act. Should be certification from the good clinical practices, certification from the shop act in Maharashtra, we know, certification with biomedical waste disposal body, no objection certificate from the fire department, and no objection certificate from the municipality corporation. These are the basic license we need to start the any particular lab into the market. So to buy the instruments is not easy. So we can raise the fund towards the government. So as a part of Infernal Biotech, here I personally invested the only 1 lakh from my pocket. So all other instruments we brought by the funding only from the government, any funding agencies. I will show in next. We will show in next slide. So we can raise the fund towards the government if you have a good idea or any particular idea to start the particular what we can say the business. So how to get fund? This is the big caution in mind. So there is some pathway you are going to raise the fund towards the government. So first, if you have an idea in biotechnology, you have to find out the biotechnology funders. You're not going to find the funders from the environmental, because if you have an idea in biotech field, you should know the who are the funders to fund us. So first, find the funders. So as per the rules and regulations of the funders, we have to develop the proposal and submit the proposal. If they are satisfied with your proposal, they will grant us. After taking a grant, you have to set up a project because there is a limitation of the time period. So you can set up a project using a particular fund from the government and manage the project. After the given time, you can close the project. This is how pathway works into the particular funding agency. So in India, who can get the fund? So these four categories easily get the fund. Research professorship like the permanent faculties, career development awards like the students from UG, PG, they can get also the funds from their business. Long-term international fellowship, the international PhD students, they can also get a fund. Project seed fundings as a business part. So I had the project seed fund. Now we completed that particular project. So we have the near 50 lakh fund from the Biotech to develop the lab, to buy the instrument, to train the students. So you can raise the business funds under the seed funding into the Biotech, also any other funding agencies. Here I listed some funding agencies which are entertaining to give the fund for the businesses. So DST, one of my favorite, Department of Biotechnology, also DRDO, BIREC, also giving the many funds to the business setup. Also ISER, some point of view. So DAE also. These are some funding agencies which are in India which are helping to set up using a particular scientific field. So how you are going to utilize the funds after getting a fund? You are not going to take into the personal, because you have to utilize that fund for the development of the research. You are going to you can use that particular fund for the purchase equipments, infrastructure, building that particular infrastructure you can build, facilities you can buy, publishing. Nowadays, the journals need a fees for the public. Distribution means if you have a group project, you can distribute within your particular group. So you can use these particular funds in this area. So Doctor, the slides are visible.
Yes, sir. Yes, sir.
So, okay, just a minute.
Only your voice is breaking, sir.
Why voice is breaking?
Yes, now it is breaking.
Yes, so some network issues going on. I'm really sorry. Just a minute. So now I'm audible.
Voice is breaking.
Voice is breaking. Just a minute. Just a minute. Just a minute. Is it clear now?
Still, there is some crack in the voice.
Just a minute. Hello.
Yes, sir.
So am I audible now?
Yes, you are audible.
Okay. Oh, those slides are visible.
No, not yet.
Oh, sorry. Just I'm really sorry, doctor, because
No
I
issues.
don't know. So
These things are.
how it's happening because some connection errors are going on from
No,
the morning. Just a minute. Give me just a minute.
these are things which are beyond our control.
Yeah, that's the thing. So I'm really sorry.
No, sir.
Think now slides are visible.
Yes.
Yeah. So sorry, audience, because due to the network errors, so we are facing a lot of problems here. So coming for the very interesting part of the research, that is R&D. So in it, we can use that particular business. We can set up a particular business using this R&D fields. So it is also plays a very important role in research and development. So as we all know, so in research and development, there are the basically five phases we are all going through. So first is a discovery and development, means we can target the particular problem and develop a proposal. We call it as a discovery and development. So some preclinical studies like in vitro, in vivo, using some cell lines, any rat, mice. We call this phase as a preclinical study. And clinical studies, as we know, we are going to try that particular drug on human beings. After this all passing the phases, we particular drug we are going to take approval from the FDA. So taking approval from the FDA, after that you can market as your products. This is a basic pathway how you can start your products or utilize your idea into the products. We have to pass these all the five phases to come out with the particular product into the market. So or this it might be take the some times because we have the all the government rules and regulations. Might be take the 4 to 7 hours depend upon the attention on the particular product. Might be also take the 4 years, 5 years depend upon the totally the interest of the researcher. So in clinical research, as we know, the main important the three phases: phase one, phase two, phase three. In phase one, we are going to test the drug on particular the limited human beings. Second phase, we are going to increase the number of the human beings. In third phase, we are going to test on particular community. So what is FDA? We all know any research products, if you want to market it, we have to take approval from the FDA. We has been the Food and Drug Administration, which is ruling from the US Department of the Health and Human Services. Under that, we have the Food and Drug Administration Department. We have to submit our drug to the government after research and take approval from the FDA. So for this, the particular R&D industries, the CGMP nowadays playing a very important role. CGMP is a particular manufacturing practices. Normally, we can call it the safety regulations which are set by the government. We can call the US FDA and WHO. So we have to follow all to ensure that whether the product is manufacturing correctly or not. We have to follow the CGMP rules also. So coming to the intellectual property laws, so if you are starting a business in private sector, if we have to take care of all our logos and all these things, for that we need a particular uh, what we can call it, because the intellectual property laws. So in this section,
Sorry to interrupt. That the screen you are
oh,
sharing has been lost.
goodness, the small slides are remained. Just a minute, doctor. I'm so sorry. Just a minute.
It's okay if you don't present. Means you can just keep the slides
Yeah,
on the
yeah,
screen.
yeah.
By presenting, it might be taking more.
More, yeah.
Power.
That's the thing going on. So I'm not understanding. The first time, like this happening.
It's okay.
Yeah,
Yeah.
so I shared you in WhatsApp. Can you share that slide on
Okay, one second.
your side? Can you please do that? So
One
I
second.
already shared in your WhatsApp.
Yes, sir. One second.
Yeah.
You can just continue. I will just share the slides.
Okay. So, yeah. So coming to the IPR part. So as we all know, so IPR, so intellectual property laws nowadays, we are facing very much problems whether the particular company stolen the logo of the particular company, any slogans. So for the whenever you're starting the company into the private sectors, we should know the how its particular IPR working into the company rules. So you can register your company logos into the IPR. We can call it as a IPR section. Also the slogans you can register under a particular IPR section. So coming towards the IPR section, as we know, so my company also at the initial stage we faced a lot of problems to select our name, because when you are got to know the people you are getting a famous people also stole some names. They will create their websites and all these things. So it will dilute your market after starting a very strong business. Your hard work also they get diluted. So IPR plays nowadays very important role to register the companies and also your logos and slogans. So can I get the slides, doctors?
1 second. I'm just sending it through mail.
Yeah. So very few slides are remained. So it will take might be the 10 minutes also. Not getting this because that's the problem.
We'll do. You can continue. So I will share the slides if required on in the group itself.
Yeah. So you can share slides. So I will just explain this one.
Okay, 1 second.
Yeah. Stop the sharing. So you can just go with the sharing.
1 second.
is it possible? Can I share, doctor? Because now I got an internet connection. Can I share it?
Yes, it's downloading actually.
Okay.
It's a big file, right? Now I can share.
Okay, okay. I'll share.
You are sharing?
No, you can share. If you
Okay.
download, you can share. Because
Okay.
now I don't know whether it's working or not.
Okay, one second.
Yeah.
Sir, in the meantime, what are the educational qualifications required to set up the plan?
Very good question. So for the set up a particular business, you should have a master's degree in your hand. If you have a master's degree, you can apply for all the license.
Is the screen visible?
No, doctor.
Not yet.
Yeah. I mean, I will share. We will see how it works.
Okay.
Yeah. Sorry, audience, because due to the internet, because it's not in my hand. I'm trying my best. Technique. Okay. It's visible, right?
Yes.
Right. Okay. Okay. So coming to the intellectual, sorry, audience. Coming to the intellectual property rights. So we'll go very fastly. The slides are moving. Yeah.
Yes, sir.
As we know the IPR, so intellectual property rights. So it will use the rights to the owner to protect their logos and slogans when you are starting the business. In that particular field, we can see the types of the particular IPR. So there are many 6 types. We can see the patent, copyright, trademark, design, database, and trade secret. So if you have idea, you can file under the patent. If you have the written material, you can file into the copyright. If you have the particular logos, you can take a trademark. If you have particular machineries designs, we can register into the design sector. Database, if you have any softwares which is helping out the research, you can register into the databases. Trade secret, the secret between the company, because we don't know whether the formulation is as we take for example of lupin and any particular example of paracetamol. We don't know the formula, how the drug becoming or how the particular drug formulated. So that we call it as a trade secret. Only company knows that things. That we call it as a trade secret. So this is how the small part of the particular intellectual property laws. So just a minute. It's again happening. So doctor, can you share? I'm so sorry. It's again happening. So you can share just in few slides are remained. Just go through it. And again, we will start the question and answer sessions. Started.
You have stopped sharing?
Okay. Just a minute.
Hope the issue is not with Zoho.
Okay. Stop. I stopped the sharing, so you can share, doctor.
Okay, one second. Actually, the issue is with Zoho.
Issue the Joe. Oh, I'm seeing the what's happening like first time.
It is. It has some error issues.
Okay.
No issues. We'll share the slides with them on the platform itself.
Okay, okay,
Continue
okay,
it. We
okay,
want to
okay,
waste time.
okay. So sorry, audience. So as a last part, so I want to introduce my company. So Infinite Biotech Institute of Research and Analytics. So located in Sangli. So we are actually dealing with the various research things like the biomedical section, animal tissue culture. Also, we have the R&D field, like the drug development and validations. So as a part of the particular Infinite Biotech field, so I'm working as a director. Also, we have the international and national collaborations. Like I also have the collaboration with the CSIR, CSR, IIT Lucknow, CSR, SIMAP Lucknow. Also, I have a very well-reputed private collaboration, Satyabama University, Chennai, also Savitri University, Chennai. And we are regularly doing restorative work in animal tissue culture. Also, we are training the students. We have the training centers. In a year, near about the thousand students, near about thousand students get getting a training into the infant biotech, like internships, OJT programs. Also, we are targeting very well reputed journals, which are like the Nature and all these things. We have the papers into that. So, coming to the research field, we are into that. So basically, we have the analytical services also. So we have the SAM, TEM, XRDs, also biomedical science like the antioxidant, anti-inflammatory assays, also some analytical, we can call it GCMS and LCMS. We have. So this is a part of small part of informatics we started in 2022. Now we are at that particular stage. We are giving analysis to the researchers. So I'm very sorry about the internet connections. So I hope you understood a little bit. I don't know. There is a very few slides I presented very well. So if any questions, you can easily discuss with me. We will. This is open for the question and answer. We have questions related on the any animal tissue culture businesses, how to start and all these things, how we are working. This is open for the all the audience. So I'm really sorry. I want to present, but there is a no right connection with me to start the slides. Yeah. Any questions from the audience?
There are a few in the chat box. You can see.
Yeah. Chat box. Yeah. Okay. So one student asking could generate AI simulated patient specific animal tissue response to the nanomedicines, accelerating formulation optimization for the targeted therapies like liposome in oncology. Yeah, we can. We can utilize the AI for the particular drug delivery systems. Nowadays in ATC, we are regularly using that. So we can use the AI system to get to know whether the particular drug working into the particular drug delivery systems. So by preparing a nano medicines or a nano formulations, we can utilize the AI in ATC. So another one question is, what are the key advantages using a primary cells versus the immortalized cells, cell lines in toxicity testing and drug screening for the pharmaceutical development? Very good question, because here is the main difference. So basically, I prefer the primary cells because primary cells have the high potency to give accurate results. So immortal cells have the low capacity to act on drug or are inhibited by the drug. So there is a two difference between the primary cells and immortalized cells. So primary cells gives a very correct results in ATGC, and immortal cells gives a very alternative or specific results we can call. Very good questions asked by the Pradipati. So Pradipati is there.
Yes, he is there.
Okay. Okay. So any question from the other audience?
Thank you very much, sir. I got the answers, sir. Thank you.
Yeah. Thank you. Thank you so much.
Before we proceed, can we just have a one photograph because we never took photographs.
Yeah.
So can we take one? All of you, can you just means you can open your means if you can on the audio
You can
the
on your
speaker
cameras and all this. Yeah.
camera. Just
Yeah.
Nobody is interested in showing their faces.
No problem.
Okay. Any more questions? Because that's what where the main part is. You need to ask questions.
Any questions from the audience?
So doctor, these are the people who are more means they would be directly involved in some animal tissue culture. So that's why they have joined the session. So
Okay.
you already know that the community has more than 900 people. So
Yeah.
depending on their interest, they join. So I guess if they have any specific questions related to their research, they can definitely uh, connect with you also. Can They can share in the group also, or they can contact me. I will pass on your number to them also.
Okay, no problem. So I'll also share the slides on your number. You can share into the group. So I'm
Yes,
really sorry. I'm not satisfied with the presentation because the internet is going on and all the things.
yes, yes.
So
Zoom
you can
has
share
some
with them.
issues, but since it is an Indian platform, we promote it. So they have their own issues. They are not up to the mark recently. Whenever
Yeah.
the load comes up, it starts malfunctioning.
Yeah, that's the problem.
Any more questions? We need more questions.
Sir,
Otherwise,
I have one question. How do
yes.
you think after the introduction of AI, will this how much impact it will have on the animal testing? Instead of labs, can it happen directly through the AI?
So, as we for the addition purpose, right now we are using a AI system because additions in most of the time in animal tissue culture we make a mistake in additions because we need a very less amount of the drugs and all things. So nowadays we are going to use the AI assisted additions. So it will help us to minimize the errors. So AI nowadays in future also AI going to work into the ATC also the also might become into R&D field. So ATC, the most of the mistakes happening in the additions only. So to overcome that problem, AI is working very good. So we are also using some AI systems to work accurately, like to microliters of the drugs, any mg or micrograms of the drugs. We are using regularly. So AI helps us to minimize the trial and errors.
Okay, I guess there are, means the question level of questions are very high with respect to AI. I
Yeah,
always ask the basic questions. So if I am allowed to ask one question,
yeah,
uh,
please.
sir, what means there are many freshers who won't ask, means they are shy or something. What do you think means what would be the basic skill if someone has to get into a animal tissue culture lab?
Yeah, yeah. So always I'm motivating the students because when I am one of the my I am one of the example because so now I am the just only the 25. So I'm the 25 because I'm motivating the students, giving example of mine because so students are lacking into the particular, they are not encouraging to ask the questions and all these things. So when I started my business in the age of the 21 only.
Hmm.
So when I'm 21, I started my business into this field. So so I suggest the students, if you have the ideas, come up with the questions, discuss with the mentors or any other friends. If you have idea, it might be also the work. So I started my particular institute with my idea only. So that's why I'm focusing on the student development because in India we have a very creative students, but they are, as you raised the question, they are shying to do the things. They're not going to ask the question. They're not going to interact with the people which there are also some many points in Indian students we are seeing. So my suggestion to the students, if you have idea, come up with the questions. If you have any particular idea, work on that. So there is no option for the hard work. If you are doing the hard work, you 100% you will get the solution on that. So I'm one of the example at the early stage. I started my business. Now I have a evaluation of near over 6 to 7 crore company at Sangli. So I have the very international strong connections, also the government connections I have till the education minister and all these things. There is nothing is impossible. You can do, but the research needs the transparency. work with the honesty in the field. If you are honest in your field, you might be at a top level at a particular research. So you can do so as a part of. I'm only the 25, so people don't think I'm the 25 because I have that courage to talk. Because the courage comes up with the hard work and the practice, also the knowledge. So I suggest students read more things on basics, focus on basics things. So also the practical skills which are most of the I think who are doing PhD, also the masters, focus on some basic skills, whatever you have. So it might be converted into some big business in upcoming days. So my suggestion to students, do some hard work, stay disciplined, and there is I am again repeating, there is no option for the hard work. Do the hard work, you will get all the results.
So there is one more question there. One was related to activity. I
Yeah.
guess if there is any activity, you can contact any of the labs. So that's not an issue. One There's one specific technical questions. Again, that AI thing has come up.
Okay. Okay. So did AI replace in vitro in vivo models? As a pharmacological pathologist and young research, we use EESP ecology. People are working on preclinical level studies. I want to pursue my career in this pharmacology. So, see, as says, there are some also the benefits of AI, some also the disadvantages of the AI. So, AI might work in upcoming days. Now, in vitro, we are using AI. So, I don't know about the in vivo. So, we are not using any AI systems in the in vivo because in vivo, there is a handling of animals and dosing of animals, collecting the particular samples from the animals. So, these all needs the skills. So there is a human being works accurately rather than the AI, I think. For that particular interpretation of data, you can use the bioinformatics tools in pharmacology. So you can pursue your career in ecology because ecology now needs a very practice and research oriented persons nowadays. Because in ecology we don't find that particular persons they are working regularly. So you can pursue. I suggest to pursue your career in ecology because ecology one of the field you are ensuring that particular drug working perfectly into the animals or not. So very good question raised by the Vijay Singh something. Yeah, yeah, very good question.
Just want to clarify, since we are having lots of AI related questions. Uh,
Yeah,
One field which would be very preserved means would be safe compared to other fields would be
yeah,
our field, pharma biotech fields,
Pharma, yeah, yeah,
because here always that human touch will be required. Whatever
yes, yeah,
experience we have, it is necessary that we, of course, you shouldn't be shy of learning AI. You should be upgraded with respect to AI. You need AI. You
yes,
accept it or not, it is the new computer systems. Means if you don't know computer, you are out. That was the
yeah,
scenario 5 to 6 years or 10 years back. But now, if you don't know AI, you would be out of the system. But that
okay,
doesn't mean that AI will eat up your jobs. Every job or whatever traditional job we have, it's just we are seeing an upgradation of the jobs. Everyone is asking about AI. Even if you are going in any section with respect to, I'm specifically talking about pharma and biotech, that wherever you go, whichever section you go, you will face AI. But just keep it in mind that you need to be upgraded with respect to AI. I
yeah,
guess almost all the courses are also getting upgraded in the at the university level also. They are bringing it in AI, Python. I can see those terms coming up in the course syllabus also.
yeah. So in our institute also we are adapting some AI technology because it speed up the work. So whenever is required, we also take some because we have to maintain the both equally. The AI also the also skilled persons. Then and then only the perfect work will be happen.
Absolutely.
Yeah, yeah.
Means you AI can't do that ELISA reading, right? Of course, you
No.
if you give that data, it will be doing some
Yeah.
interpretation, but
Yes.
it cannot come and read your results.
Yes. Very true.
Yes, any more questions? Otherwise, I guess one from an entrepreneurship point of view, just if one what was the one challenge which you faced, since you are a very young entrepreneur compared to me. I am in my early 40s, so you are an early person. So, what was that one challenge which you faced, which you can share with these upcoming entrepreneurs?
Okay, so earlier I faced one challenge. So I'm sharing it very personally because earlier I faced the challenge regarding the licensing. So main problem I faced because the government is not allowing at the early stages to start the business in the R&Ds. That's one major problem I face. Second, the people's at the early stage see at you, so they will ask the qualifications. That is a major thing. So, but I have knowledge. I came up with all the things, and the another one thing is the knowledge. So when I started the business, I had the only the basic knowledge. When I came up with some questions with the in front of my feet, the doctorate and all these things, I upgraded myself. So the main problem I faced in particular the entrepreneurship is the licensing. In India, we have to go through the every documentary and all things. So that's the thing. Also, the skills matters. So I developed my skills after starting the business. So early stage, I had a small skills and all these things. The major problem in India to start the business is the licensing part. So everyone get to know how it's work and all these things. So now it's all totally online, but we have to face the panel and all these things. So that's the major problem issue I faced while starting my business.
Okay, so if there are no more questions, we'll stop for the day. I'm really thankful, Doctor, for accepting our invitation and coming up with this topic. And I'm very happy that you covered right from your basics to the means right from theoretical knowledge to setting up a lab. So that's not easy. You have shared it well, in spite of all the issues that we faced. We just a request to the audience, if for your certificate's sake, please drop a message with your name to me personally. You can't message in the group. I'll share the certificates with you. Also the slides that sir was presenting, I'll be sharing it in the to you personally, not in the group, because it's a big group. So I'll be sharing to those who will contact me directly with respect to the certificate part. Okay. So, sir, if you want to say anything, otherwise we will break for the day.
So if the audience want to contact me, so you can share my number and mail ID to themselves, so they can easily get to know so how it's working and all these things.
Okay.
So again, I really sorry because I want to express more due to this internet connection. I'm not able to present well because most probably I'm very honest. I like physical presentation rather than the online because online like this thing happens. But I'm really sorry for the audience and organizers for this inconvenience. So we hope we will build a very good research India in future. So thank you so much for your valuable time and thank you, Doctor, for the opportunity to interact with these young minds. Thank you so much.
Thank you so much. Have a good day. Bye.
Thank you so much. Thank you.